All right, let's dive in. Today, we're going to pull back the curtain on forensic DNA. We're going to follow the incredible journey from a tiny speck of biological evidence at a crime scene all the way to the powerful digital data that can change everything in an investigation. Let's get to it.
So take a look at this graph. With all its colorful spikes, this thing is called an electrophoredogram, or EPG for short. It's so precise it can literally identify someone from a single cell. The big question is, how on earth do scientists get from a swab at a crime scene to this exact digital fingerprint? Well, that's exactly what we're about to break down.
You see, for a forensic analyst, that graph, that digital fingerprint, is everything. But understanding it isn't just about reading the final result. It's about knowing the entire story of how it was made, from start to finish. That knowledge is what allows them to tell the difference between real genetic information and just technical noise.
So it all starts way before we see any of those flashy peaks. The raw evidence has to be prepared with incredible care in the lab. Everything that follows depends on getting this first part right. It's a step-by-step workflow that turns that physical sample into a pure analyzable signal.
This whole process really boils down to three key stages. First up is extraction. That's literally just breaking open the cells to get the raw DNA out. Then comes quantitation, which is super important. You have to measure exactly how much DNA you're working with. Too much or too little can mess up the whole thing. And finally, amplification. This is where we make millions of copies of just the parts of the DNA we need to look at.
Now, that third step, amplification, is where the magic really happens. It's all thanks to a technology called PCR, the polymerase chain reaction. The easiest way to think about it is like a super-specific genetic photocopier. It doesn't copy everything, just very particular spots in our DNA called short tandem repeats, or STRs. And it doesn't just make a few copies, it makes millions, even billions of them.
And here's the really clever part. While that genetic photocopier is running, it's also attaching tiny fluorescent dye tags to every single copy it makes. Each tag is a different color. You've got blue, green, yellow, and red. This is the absolute key, because it's what turns these invisible DNA molecules into something a machine can actually see and measure.
Okay, so now we've got our evidence, and we've made millions of fluorescently tagged copies. So, what's next? Well, we send them to the races. This is where a machine called a genetic analyzer takes over, using a process I like to call the DNA racetrack, or as scientists call it, capillary electrophoresis.
So what is capillary electrophoresis, really? It's a super cool method for sorting DNA fragments based purely on their size. See, DNA has a natural negative charge, so if you apply an electric current, you can pull it through this incredibly thin glass tube, that's the capillary, which is filled with a thick, gel-like substance called a polymer.
Think of that gel as a molecular obstacle course. It's a dense matrix, and the DNA has to wiggle its way through. The smaller, nimbler DNA fragments can snake through it really fast, but the big, bulky fragments, they get tangled up and move much, much slower. So just like in a real race, the little guys always win. They cross the finish line first. Now, at the very end of this tiny racetrack, there's a detection window. And as each DNA fragment zips past the finish line, a laser zaps it. That zap makes its little fluorescent tag light up and glow, and a super sensitive camera is sitting right there recording the exact color and the brightness of that flash of light. This is the moment. This is where biology officially becomes digital data. But there's one little hiccup the system has to handle. Sometimes the light from the different color dyes can kind of bleed into each other. You know, the glow from a blue dye might spill over a little bit into the green sensor? So the software has a built-in color corrector called a spectral calibration matrix that digitally unscrambles all the signals to make sure every single color is read perfectly.
Okay, the machine has done its job. It's captured all those tiny flashes of light as raw data. Now we get to the final and maybe most important phase, actually decoding that data. Let's see how the software translates those light signals into the meaningful peaks on our graph.
The graph is actually pretty simple when you break it down. It has two axes that tell us two different things. The horizontal one, the x-axis, tells us the size of the DNA fragment. It answers the question, what is it? Then you have the vertical one, the y-axis. That one tells us the amount of DNA. It answers how much of it is there. That's it. What and how much. So you might be wondering, how does the machine know the exact size? It doesn't have tiny eyes to count the base pairs. It needs a ruler. And that's why in every single run, scientists add something called an internal size standard, or ISS.
It's just a mix of DNA fragments of known sizes, all tagged with a specific color. The software sees those known points and uses them to build a perfect ruler to measure all the unknown fragments in the sample.
Okay, so now we have a size, but a size is just a number. It needs a name. For that, the software uses an allelic ladder. Honestly, the best way to think about it is like an identity lineup for DNA. The ladder contains all the common known alleles. The software creates these little digital bins or windows around each known allele. Then it just looks to see which bin our sample's peak falls into. If it lands in the allele 14 bin, boom, that's its name.
So what about that y-axis, the height of the peak? Well, that's measured in something called relative fluorescence units, or RFUs. Don't let the fancy name fool you. It's a really simple idea. The more DNA fragments of a certain size that cross the detector, the brighter the light they give off and the taller the peak gets on the graph. A higher RFU just means there was more of that specific DNA.
But you can have too much of a good thing. If there's way too much DNA, the signal gets so bright that it completely overwhelms the camera's detector. When that happens, the peak gets its top chopped off. It looks flat. And that's a huge problem, because it means you've lost all the quantitative data. You can't tell how much DNA was really there. This is why that quantitation step way back at the beginning is so incredibly important.
So we have our graph. All the peaks are sized and named, but we're not done yet. Now comes the human element. The final, critical job for the analyst is to go through and make sure the whole profile is accurate, that they're separating the true genetic signal from all the potential background noise. So, what are they looking for? Well, the gold standard, the ideal peak, is this: a clean, sharp, perfectly symmetrical bell curve shape. It's what mathematicians call a Gaussian distribution. When an analyst sees this, they know the process worked beautifully for that piece of DNA.
But science isn't always that perfect. Sometimes little technical glitches happen, and they create things called artifacts. A really common one is what you see here, often called a split peak or stutter. This is usually caused by a tiny hiccup back in that PCR copying step. It can make one real piece of DNA look like two little stuttering peaks right next to each other.
And this is where the skill of a forensic DNA analyst really shines. A huge part of their job is being a data detective. They are trained for years to spot these technical artifacts and filter them out, making absolutely sure that only the true, reliable genetic data is used in the final report.
So let's just recap that entire journey, because it is pretty amazing. We went from a biological sample to making millions of fluorescent copies of its DNA, sending those copies on a high-speed race down a tiny tube, turning them into flashes of color light, and finally, translating all of that into a precise digital graph.
You know, the source material we're looking at sums it all up perfectly with this quote. The EPG is the culmination of a linear analytical process transforming biological material into digital data. It really is a mind-blowing transformation from the physical world into the digital one.
So, the profile is built, it's been quality checked, and now we have a clear genetic fingerprint from our evidence. But I'll leave you with this final thought. What happens when that evidence has DNA from 2, 3, or even more people all mixed together? Well, that's when the real detective work begins.